on the effects of knocking down raptor on mTOR1 activity and hepatitis B virus replication. So why is hepatitis B virus important? Around the world, 254 million people are infected with hepatitis B virus, and there is a yearly increase of 1.5 million. On top of that, there are about 1.1 million deaths related to hepatitis B virus illnesses, such as liver cancer. There are treatments available, but there's no direct cure. So this is the HBV life cycle. The HBV virus attaches to the liver cell, which then goes through endocytosis, and the membranes fuse together, leading to a nucleocapsid release. After that, the nucleocapsid attaches to the nucleus and goes to genome repair and transcription, where certain proteins are expressed. If you look down there, the green one, that's called HBS, and it's very important for my research. After that, these proteins go through re-encapsulation and go through reverse transcription and leave the cell to infect another cell. So, talking about the HBX protein, it's a small regulatory protein encoded by HBV, and it interacts with a vast array of host proteins. It's very important to my research specifically, because it influences cellular signaling pathways, which is what mTOR1 is. Cellular signaling pathways are just how cells talk to each other. So this is what mTOR looks like, and if you remember from the title slide, we had mentioned raptor, which is a part of the signaling pathway. The other picture shows how if mTOR is activated, so is the S6 protein. So prior research shows why is mTOR important to HBV replication? Prior research shows that mTOR inhibits HBV replication. As you can see in the DMSO sample, that there was not any mTOR inhibitor, so there was more mTOR present, and when you check the DMSO on the other side, there was not much HBV replication shown. But in the rapamycin, there is a mTOR inhibitor, leading to less mTOR and leading to more HBV replication. And the opposite is also true. If you stimulate mTOR, HBV replication decreases. As you can see, compound C is a stimulator of mTOR, and HBV replication was lowered. So my hypothesis was, if you knock down raptor, which is a part of the mTOR pathway, it would result in a decrease of HBV, because if you knock down a protein in the pathway, it should also knock down the pathway and lead to less or more HBV. And this is a visual of what we're doing. We're basically trying to get rid of the raptor in the mTOR signaling. I had done 3 processes to determine my conclusion, and the first one was a Western blot, where I had a cell culture that I had infected with the virus. Then I extracted with protein by running it on a vortex, which is basically just spinning it fast, and ran it on a gel electrophoresis. After that, you can see the protein separation based on their size. From there, we got the gel set up and transferred through electroblotting to get the proteins on a membrane. From there, we got those membranes and then washed them through 2 antibiotic cycles, a primary and secondary, and we could visualize them on a machine. So this was my practice and Western blots. As you can see, there are many proteins there. An important one to look at is actin, which is just a sample, and it shows how much of your sample is loaded. And as you can see, my actin 3 band looks a little wonky, and this is to show that mistakes do happen, but it did get better over time as my actin 2 band seemed more stable, but the first column was lowered, and that is also shown in my PS6 and core protein, where the first column seems to have less protein expression, and my actin 1 band seems to be the most stable, which is also shown in my S6 protein, where all the protein expression was also stable. So the second process I had done was an HBV replication assay, where my cells were, again, treated with the HBV virus and running through a drug treatment. From there, we had lysed those cells and put them under Bradford and treated them with DNAs and RNAs. After that, we centrifuged them, which is, again, spinning it really fast for 3 hours, and as a result of that, we had gotten an isolated nucleocapsid. Those nucleocapsids we had ran on an agar straw to visualize the nucleocapsid levels, and Then we further transferred it to a membrane so that we could denature them and probe for rcDNA. rcDNA shows how much HBV replication was present. So these are my practice, native Western and Southern, and the primary goal for me was to see if I could get the samples loaded on there pretty evenly, and the samples were pretty stable, and you can see in the rcDNA that they do correlate with each other. The last technique I had done was a transfection technique, which is to see how I would transfect my cells with the virus itself, and what would be the most efficient way. So we had 4 different concentrations, and it was 1.0, 2.0, 3.0, and 4.0. And we had learned that 4.0 was the best transfection concentration for me to infect with my cells and have the most of them affected by the virus. So coming back to my hypothesis, we were questioning, did SH1 raptor effect have an effect on HBV replication? So as you can see on my left, this is my Western blot, and we were trying to see the raptor knockdown. So we had tested the raptor, and these 2 cells are my cells without the virus in them, and the next 2 do have the virus in them. So we had to see if the raptor was actually knocked down, and we were not able to come to a conclusion based on these samples if the raptor was knocked down. So we had tested the PS6 to see if mTOR was knocked down, and we could not tell that either. So as a result, this was kind of an inconclusion, and we were not able to tell if it had an effect on the HBV replication. But we tested SH2 raptors, and SH2 raptor, the raptor levels seemed to be knocked down, as we had quantified these data to make sure that the samples were actually lowered, and you can tell that in our control and our HBV control that the raptor did go down, and the same thing with our PGEV and our HBV raptor. The raptor concentration did go down, which also led to a increase in HBV replication. So what were my conclusions? SH1 did not effectively knock down raptor, but SH2 raptor did effectively knock it down, and that resulted in an increase of HBV replication. I've also learned 3 major techniques that help me determine this. I would like to thank the Medical University of South Carolina, the Bouchard Lab with Dr. Michael Bouchard, Cody McCumber, and Kendall Torres, and the Governor's School for Science and Mathematics, Dr. Josh Witten, and Dr. Lance Riddle. Thank you.